Hello and welcome to this ViBrANT talk, and today we will discuss with Maria and Juan about the biosensors, because in both their projects they are using or developing biosensors for diagnostics purpose. But what exactly is a biosensor, Juan?
Well, a biosensor can be defined as an analytical device, which is composed by three main elements. First element we will say is the bioreceptor, which normally is they have the molecules, the biomolecules, or even entire cells. Their function is to capture the analyte. So, for instance, the most common bioreceptor in biosensors antibodies or derived molecule from antibodies, but also nucleic acids, and as I said before, entire cells. Yeah. So then we have the transducer, the transducer element. Basically, the transducer element is the way that the biological binding is interpreted, right? So we can find three main different transducers, which normally defines the type of biosensor, typically optical, mechanical, and electrical. It depends on the classifications that are different, but normally this three. And then we have the signal display, which tends to be either in a computer or others just on the screen. And nowadays, it is more typical to be connected on phones as well. Yeah.
Okay, so now we have the definition in place. So which kind of biosensors did you develop, Maria, during your PhD?
Hi, Thais. I developed, or I am developing, two biosensors to detect two different kinds of bacteria which cause infections in human beings. One of them is Yersinia enterocolytica, and the other is called Moraxella catarrhalis. And my biosensor, so there has to be a way for which the biosensor is able to identify this bacteria and not other bacteria. So how we do that is we use a bioreceptor, or in simple words, it's anything that has an affinity for that particular bacteria or a part of that bacteria. So I am not working with the whole bacteria, but with a specific protein on the surface of each of these bacteria. So the one specific bacteria on the surface of Yersinia is called Yade, and the specific bacteria that I'm trying to detect on Moraxella catarrhalis is called USP82. So what is the bioreceptor that I'm using? It's a piece of DNA sequence. I guess everyone knows what DNA is, but I'm just using, so it's possible that just a piece of DNA sequence can sometimes have affinity for this specific protein. And I use a process from which there is a group of millions of these sequences called aptamers. And I used a process called SELEX, to expand SELEX, it's systematic evolution of ligands by exponential enrichment. Okay, I know that sounds complicated, but what I did was from this group of millions of sequences, I was able to, through this method called select one or two sequences or aptamers which had high affinity for these proteins I was trying to detect. So once I had my specific aptamer or sequence which has high affinity for the protein I'm trying to detect, I took my sensor, electrochemical sensor, and I immobilized this sequence. I made a lot of the sequences and I immobilized them on top of the sensor. And then in the lab to validate or to see that if it actually has affinity for these proteins, I just dunked a whole lot of the protein on top of the sensor. And then before and after this process, so before dunking all those the proteins I'm trying to detect, I measured the signal. So if there's a major change in the signal before and after, you can, in very simple terms, conclude that my sensor works because there is a change in the signal. Of course, it's more complicated than this and nothing works perfectly, but just to understand the principle of how it works.
That sounds very interesting, and sounds like an easy method to detect the bacteria in. So Juan, did you have the same approach, or did you do something differently with your biosensor?
Well, my biosensor, what I'm trying to detect is Yersinia enterocolytica, but in my case, it's whole cell. So what I'm trying is, on the one hand, expressing, because so from the beginning, I'm not trying to detect Yersinia enterocolytica. So what we're doing is to express one protein, which is Yersinia enterocolytica A adenosine, or YAT-A. So we express this protein on on E. coli to work with it for security reasons. So basically, this adenosine YAT-A is involved in the first step infection of Yersinia. What we're doing is to express this protein in E. coli and observe whether there's binding or not to an extracellular matrix protein, which this YAT-A has affinity, which in this case we use collagen. Set that, my biosensor consists in coating surface with collagen and see whether these E. coli that have expressed YAT-A, they bind or not to the collagen. And then as a control, we bind E. coli without any expression of YAT-A at all. If we go in detail about what my biosensor is in terms of optical, mechanical, and stuff like this, my biosensor is electrochemical. And particularly, I use a technique which is called electrochemical impedance spectroscopy. So what we do is to measure the presence of something attached to the collagen surface, which in this case is the bacteria, by measuring the impedance, which is typically like a resistance found on the system. What we measure is in presence of bacteria. So before the bacteria, we measure the system, and then we measure the system again after the bacteria. Then, by comparing the system, like the signals before and after, we will know whether we have bacteria or not. And also for that, we need controls as well, which is the bacteria without expressing YAT-A. And also as well, we have controls without any bacteria, just to see how the system itself, which can be the buffer, can change the signal as well. So we have to work with all those factors. This is how my system works in principle. Yes. So far, I can say that we've been successful in detecting the whole bacteria, but still we have to work in the limit of detection. It's still very high. Even more if we compare to the classic biosensor using antibodies.
Now we heard about your biosensors, but maybe it would also be interesting to hear a bit about your point of view on using biosensors as a diagnostic tool in the field of diagnostics in the hospitals. Which advantages does it have, and which disadvantages does it have to use biosensors? Maria, I don't know if you can enlighten us on this.
Sure, Thijs. One immediate major advantage of biosensors is that they are portable and they are produced cheaply. So they can be used, you don't have to go to a lab to get tested. You can do it at home, as we've seen with the pandemic, in a restaurant. And so the focus, you said my biosensor sounded so simple, because the goal of my work was to make a cheap and simple biosensor so it can reach people who cannot pay for an expensive lab test. So those are really the main advantages. Of course, the disadvantages can be that maybe some biosensors may not be as sensitive or as some very high-end equipment in the lab. But that is also not true, because we are developing biosensors now which have very low detection limit, and which in other words means they are very sensitive and specific. So there are a lot of advantages to using biosensors, and I think because of the low cost paradigm, you will see them being used a lot more and more.
And do you think that biosensors will replace the current methods of diagnosing different diseases, or do you think that we still need to use both strategies in the future?
From my perspective, I think if biosensors ever take over techniques such as ELISA, PCRs, and etc., I think it's not going to be in the short term. I think it's going to take it in decades. I think biosensors is relatively new, relatively, of course it's not, but I think it's a very mature science compared to those techniques. And I think we are on a good path. We are doing well in terms of developing and achieving good limit detections. There's a long way to get there. So I don't know, and I don't have the courage enough to say whether we, like biosensors, are going to take over those techniques. But what I know for sure it's not going to be now. It's going to take a while. For instance, in this pandemic, I think people have realized about the importance of diagnostic techniques. I don't think the target is to take over the current diagnostic techniques. I think it's there to help. They can be used for rapid screening. They can be used for many things, not just. So far, I think that ELISA, PCRs, and other techniques, they are very specific. They're very expensive as well. They're very specific, and so far, probably, if we compare that to a current, like the state of the art of biosensors, we cannot achieve that yet, at least in general terms. But for sure, if you want a rapid screening and you don't need that precise signal, maybe biosensor can provide that. So I think at least, if not the future, part of the future. Yeah.
I want to reiterate the point also that Juan made. It's not about replacing the kind of lab techniques and the ones that already exist, but it's about which system works better in a particular situation. So we are going to try to use biosensors together with existing lab techniques to make the system better.
And so from like the example I think of immediately is like Juan also said about like during the pandemic, everybody knows about the antigenic test, but often you want to validate the actual positive case with a PCR test. So here we see the interaction between the biosensor and also the more traditional technique that takes longer time to develop. So I think everybody nowadays can see the usefulness of these biosensors.
Exactly, that's a good example. Yeah.
Now, I'd like to add something which I think it is very important, at least in my type of biosensors, but can be not only in the biosensor I'm using, but biosensors compared to more specific techniques. Sometimes biosensors compared to traditional techniques. I mean, traditional techniques can offer as well what I'm going to say, which is kind of specificity. Sometimes you want to detect something, but you don't need to be that specific. It's going to take longer, and if it's going to be very expensive. Sometimes you just want to do a rapid screening to know a family of organisms there to start a treatment. And then once you start the treatment, you can take more time to then know which specific organism it was, right? So I think biosensors can offer that as well, like rapid screening that, for instance, a clinician can do while you are there. Rapid takes minutes and can say, well, this is a family of these organisms, we can start a treatment with this. And then you can wait for a more specific technique to say, well, that was that organism. And then you can not start, but you can continue with the treatment or not. But at least you had that screening. Like, that's what we said, like, biosensors are not there to replace, are there to as another tool of diagnostics.
Yeah, so you can faster have an idea of what infection the patient have, and then thereby get to know faster what treatment the patient needs to get. So I would like to thank you both for participating in this talk and enlightening us all on some other biosensors than the antigenic COVID test. So thank you, Maria and Juan.
Thanks, Thijs and Juan.